Our final installment for Chapter 9 involves anaerobic respiration, something that we call fermentation. And there are different kinds of fermentation, including fermentation or anaerobic respiration that we do in our own bodies. So it's a very, very important source of immediate energy for things like maximal contraction and power and so forth, but also very, very important in the industrial world, in groceries and in food products, and in microbiology. Given that we don't really operate at levels of muscular contraction that are anaerobic, it stands to reason that most of our ATP is produced oxidatively when our muscles are not contracting all that much. And that requires oxygen and requires blood flow and lots of things going right. However, if we do get to the point where there isn't enough oxygen, the electron transport chain won't function, and we will actually shunt our pyruvate into lactic acid until oxygen returns, and then we will go back to the oxidative side of it. But it actually happens surprisingly often that we switch to an anaerobic condition. And when we switch to an anaerobic condition, what we're going to do is we're going to use glycolysis to create pyruvate. And so here we are splitting the glucose into 2,3-carbon pyruvates, and then those pyruvates will then be converted into lactic acid. Another way of putting this is saying that we're going to use a different final electron acceptor than oxygen. And part of that final electronic accepting involves the creation of something like lactic acid or other types of products in other types of fermentation. To this point, we've talked about aerobic and anaerobic situations. There is a little bit of an aside, and I just want to bring this up a little bit, is that there are forms of anaerobic respiration outside of fermentation, which can use other electron acceptors. For instance, bacteria and certain types of fauna that live in volcanic vents that are very rich in sulfates, or hot springs like in Yellowstone. They can do an equivalent of aerobic respiration using an electron transport chain. It's just that oxygen isn't going to be the final electron acceptor. Sulfate and other types of molecules become the acceptor, and so it doesn't need oxygen, and that's why it would be anaerobic. But for our purposes, we're going to just kind of generalize that we have aerobic over here in the mitochondria, oxidative phosphorylation, and then we have fermentation, which can either be ethanol or lactic acid, and that's the common situation for 99% of life on Earth. But I did want you to know that there are other forms of anaerobic respiration out there. But that fermentation, either ethanolic or lactic acid, instead of using that true electron transport chain that we saw in oxidative phosphorylation, they use substrate level phosphorylation like we talked about earlier in this chapter. We're going to use enzymes to transfer phosphate off onto ADP and make ATP. So here's a figure from your book showing that we start in the cytosol with glycolysis. We bring that glucose into the cytosol, we oxidize it into pyruvate, and then if there's oxygen present, we head off into the mitochondrion to make acetyl-CoA and then through the citric acid cycle. Or we stay in the cytosol and we reduce the final electron acceptors using NAD to make either ethanol or lactate, depending on the organism and the enzymes. Two things that fermentation has in common with oxidative phosphorylation. Both involve glycolysis, generating pyruvate, and both use NAD. NAD is used slightly differently in fermentation than it is in oxidative phosphorylation, but it is still an electron carrier. It's just that it's transferring its electrons in a different direction than into the oxidative phosphorylation electron transport. When we have two flavors of fermentation, we have alcohol fermentation, so your beer and wine, that sort of thing, that makes ethanol is one of the common products there. The other one is called lactic acid fermentation. And lactic acid fermentation is very important for things like cheeses and yogurts, those sorts of types of foods. They make lactic acid rather than ethanol. In ethanol fermentation, alcohol fermentation, we're converting pyruvate to ethanol. Pyruvate's a 3-carbon molecule, and ethanol is a 2-carbon molecule. It's 2 steps, and we're going to release one of those carbons into CO2. Yeast, which is a eukaryote, is often used in this situation because it has the metabolic machinery for doing that 2-step reaction. Here are the overviews of alcohol fermentation and lactic acid fermentation. The main thing that I want to show here is that during glycolysis, and I didn't emphasize it probably as much as I should have, we're going to make a little bit of ATP, and that's going to be useful. And we're also going to be cycling NAD and NADH. I didn't really emphasize it all that much, but we reduce a little bit of NAD when we do glycolysis. And that will actually help with the second step of alcohol fermentation and the major step in lactic acid. So when we get to pyruvate, if we can't go into citric acid cycle, what will happen is that electron carrier will then assist in creating either ethanol in the alcohol fermentation or lactic acid in the lactic one. But let's dig just a little bit deeper, just a little bit. As I said before, the glycolysis is the same. That's occurring in the cytosol. We're making a little bit of ATP. There's an energy investment and then an energy recovery, and we net 2 ATP. We also net a little bit of NADH. We're going to enzymatically convert pyruvate into this intermediate acetaldehyde, okay? And we're going to create some CO2 with that, and then that NADH is going to reduce that acetaldehyde into ethanol. So that's ethanol fermentation. It's a 2-step process. We make a little bit of ATP here, and we're going to use that NADH to finalize our reduction of electrons to ethanol. Now, lactic acid fermentation is essentially a 1-step process, a 1-step reduction. But what's really cool about it is that it doesn't release CO2. Unlike ethanol production, you're not making carbon dioxide. You're keeping all the carbons that you had in pyruvate, or it says pyruvic acid. Pyruvate and pyruvic acid are the same thing. We're just using NADH to reduce pyruvic acid using a different type of enzyme system to make lactic acid or lactate. Some fungi can do this, cheese and yogurt can do this, and your skeletal muscles can do this in your body. You ever hear that phrase, feel the burn? Well, that burn comes from the buildup of lactic acid in your skeletal muscles. So we use the lactic acid to generate ATP when we don't have enough oxygen to meet all of our ATP demands. Again, it's very, very fast. It's very fast. When you go into full power, and I will show you a picture in the next slide of this, when you go into maximal exertion, you basically go straight into lactic acid fermentation in your skeletal muscle. Turns out the fibers in your skeletal muscle have different specialties, and some are more able to do lactic acid, and some are more able to do oxidative phosphorylation. And training can actually adjust the level of ability of these muscle fibers. But for our purposes, the purple ones are fast twitch fibers. The orange ones are medium, and the green ones are slow twitch. The medium ones are the ones that can kind of flip from slow twitch to fast twitch a little bit. But in any case, these fast twitch fibers can generate a lot of power very, very quickly, but they do so by generating ATP with via lactic acid. So if we look at how much tension we apply, if we apply more and more force in our muscles, when we're applying a little bit of force, we're using ATP oxidatively. But when we start flipping into stronger and more power, we become more and more lactic acid oriented in our muscle contraction. And so that's why we want to learn about this, because we can understand power and athletic performance by understanding a little bit about fermentation. So again, the big difference between lactic acid fermentation and ethanol is that ethanolic fermentation is that you start with your pyruvate and it's a one-step process, and you have a different enzyme here. I should probably draw an enzyme right here. We're still using NADH to reduce that pyruvate, but we are making lactic acid rather than ethanol. Ethanol is a two-step process that liberates CO2. So we have a smaller molecule called acetaldehyde and a different enzyme that uses NADH to transfer those electrons on there and becomes the final electron acceptor. So when we compare fermentation with anaerobic and aerobic respiration, everybody uses glycolysis. It's very ancient. It's very, very ancient. We are creating ATP, and this is pretty fast. This happens fairly quickly. All three situations, aerobic, anaerobic, fermentation, all use NAD as the oxidizing agent that carries electrons. But they have different final electron acceptors. Okay, oxidative certainly uses oxygen. Fermentation uses an organic molecule, either leading to lactic acid or ethanol. And then true anaerobic respiration, which is again relatively rare, may use some other molecule as a final electron acceptor, but it's usually like sulfur of some kind. Cellular respiration is the most efficient. You know, it can produce somewhere between 30 and 34, maybe 36, sometimes maybe 28 ATP. Fermentation is not nearly this great, and that's why you don't see giant yeast balls, yeast organisms around. You don't see things that live on fermentation. Probably the single biggest entity that survives on fermentation might be a tumor in cancer, okay, a solid tumor. They can get by, but they have massive blood supply. They're very, very good at recruiting and developing blood vessels into them. In any case, it's not very efficient, but it is quick. We can classify microorganisms. We can even classify our cells in the way that we handle oxygen or can create ATP anaerobically. First of all, let's look at tube number 2 here. Tube number 2, and by the way, each of these tubes is a different organism growing in media. Tube number 2, the cells can only grow way down deep in the tube. They only seem to do well far away from oxygen. That means that oxygen is actually a toxin in this case for these cells. And so these cells are obligated to be anaerobic, and that's where the word obligate anaerobes come from. If you're familiar with different types of beer, we talk about ales and lagers. Number 2 here would be the type of yeast that a lager is made out of. It's made out of an obligate anaerobe. It gives it a slightly different flavor profile. Many yeast and other bacteria are what we call facultative. That is to say, rather than being able to live exclusively in anaerobic conditions, they can kind of do both. So tubes 3 and 5 here are what we would call facultative anaerobes. They can grow in oxygen, and it looks like 3 does a little bit better in oxygen. 5 is a true facultative anaerobe. It doesn't seem to care if there's oxygen or not. In those cases, they can switch. They have the ability to switch, and they switch at the level of pyruvate. So if we go back to look at ethanolic or lactic fermentation, that we always make pyruvate, and it's in that decision that we either are going to go aerobically if there's oxygen, or we're going to go anaerobically if there isn't. This is what your cells, most of your cells do. Now, neural cells can't do this, and cardiac cells can't do this, and kidney can't do it, but most cells in your body are facultative anaerobes to one degree or another. They can switch to an anaerobic ATP synthesis if needed. Factoids about glycolysis is that we think it's very, very ancient, even before oxygen was available. And oxygen really didn't start showing up until we started seeing a fair amount of photosynthesis. So it's been around for a while, certainly. 2.7 billion years is nothing to shrug at. But what was happening was that oxygen was rising to the point where it could be potentially toxic. And so prokaryotes were just fine doing glycolysis until the point where oxygen concentrations got so high that in order to survive, they had to learn how to use it. So glycolysis is a very, very ancient process. Our most ancient fossils and so forth have signs that they underwent glycolysis. And connecting glycolysis and citric acid cycle, they don't just feed into electron transport or fermentation, they literally feed into almost all of the metabolic processes of the cell. Right here, this circle right here, it's actually going this way. This circle right here is citric acid. But you can see, if you look really closely, you can see that these lines connect to all different areas of the body. It's part of a larger, what we call metabolic continuum, where glycolysis is, I'm guessing, is probably right in here. It could be over here. It's hard to see. But in any case, both of these processes, one is a catabolic process. Another one is kind of a combination of catabolic and anabolic processes, set us up for precursors for all kinds of molecules around the cell. Cell is incredibly elegant that way in that it reuses things for different purposes. That's why certain nucleic acids can be converted into precursors for citric acid cycle. Certain amino acids from proteins can be either turned into acetyl-CoA or enter the citric acid cycle. Certainly sugars, we talk about glucose being our poster child. They go through pyruvate and acetyl-CoA that way. And then finally lipids and fats. When we oxidize fats, when we're burning fats, we're making acetyl-CoA in a process called beta oxidation. When we break down fats, what we do is we clip off the fatty acids from the glycerol. This is one of the reasons why I had this question in exam 1, for you to understand what makes up these biomolecules, because it plays right into this slide and further down the line in this class. So we clip off the fatty acid from the glycerol. The glycerol can be used in glycolysis if needed, or it can be recycled. And then we oxidize fatty acids by what's called beta oxidation to yield a 2-carbon molecule, acetyl, and then link it to CoA. And you can generate a lot of energy from this because you have lots and lots of covalent bonds. Now, what do we mean by beta oxidation? If we look at a hydrocarbon, or fatty acid in this case, we see right here this first carbon after the COOH. This first carbon is the alpha carbon, the second carbon is the beta and the gamma and so forth, and we go all the way to the omega. You ever hear of omega-3 fatty acids? That's because at the third carbon from the end, so omega -3, we have a double bond, and that gives it a little kink. Okay, this is a nice saturated fatty acid, but if we had a double bond there, it would add a kink, and that changes its chemical properties, much healthier than a saturated fatty acid. In any case, we oxidize between the beta and the gamma, and that's where beta oxidation comes from. So there's the alpha and the beta. Omega would be down here. So we're going to oxidize at the beta to create the acetyl group in acetyl-CoA. This chart right here shows how it's all connected. Our poster child mechanism is right down the middle, starting with glucose going this way. But other sugars, if they're other than glucose, may enter the glycolysis pathway through a different intermediate in order to get us to pyruvate. Fats, as we just mentioned, fatty acids, we beta oxidize, and that makes acetyl-CoA. Or we can take glycerol and, just like a non-standard sugar, enter it into the glycolysis pathway, and then we get to pyruvate. Proteins, what we do is we digest them into amino acids, and then depending on the kind of amino acid, and that's the R group, again, another question that we saw in the exam, the different amino acids differ in their R groups. So depending on what R group you have, maybe you can convert it to pyruvate, or you can convert it to acetyl-CoA, or you can convert it into something that's an intermediate in citric acid cycle. That's why you can enter at the 3 levels. It depends on the R group on the amino acid. But it's all connected. It's all connected, and that's what I think makes it fascinating, is how the body is able to switch and adapt depending on what kind of calories you're getting, what kind of nutrients you're getting. You can adapt this for almost any situation. Connectedness not only is involved in catabolic pathways, getting us to acetyl-CoA or some intermediate in the citric acid cycle, but it also plays a role in our anabolic pathways. We turn complex things into simpler things through catabolic pathways, sometimes releasing energy, sometimes releasing heat, almost always. Nevertheless, those building blocks we then use to build our macromolecules using some of the energy that we've harvested from the catabolic pathways. Go back to this scary picture for a second. As complicated and as awesome as this is, if all of these reactions were going on simultaneously, that would be a disaster. These are actually all the reactions that could go on in the cell that we know of, but they don't all go off at once. The reason being is that the cell doesn't have enough energy, doesn't have enough nutrients and building blocks to run all these reactions simultaneously. Even our paradigm here, why would we break up proteins that are important for our structures and other reactions in our bodies if we have plenty of carbohydrates and fats to use? So even something that is relatively simple compared to that previous picture, we have to have ways of turning these pathways on and off. Turning them on when we need them, turning them off when we have better alternatives. And so we're going to talk a little bit about that idea of regulating, and we're going to talk about regulating cellular respiration. The most common mechanism for these types of pathways, and in that first picture that I showed you, is called feedback inhibition. It's a form of negative feedback. That is to say, when we create a product, when something is created, that product then will inhibit earlier steps in the process of making that product. It becomes an inhibitor of maybe the first enzyme in the process. Cholesterol does this, by the way. If you have enough cholesterol in your body, the cholesterol will actually inhibit the first enzyme in this pathway. Lots of things do this, lots of pathways where the end product serves as an inhibitor of earlier stages, oftentimes the very first step. For instance, if ATP begins to drop, the process of making ATP will speed up. When there's a lot of ATP around, the ATP will act as an inhibitor and slow down the creation of additional ATP. It will slow down glycolysis and citric acid cycle. And that's how we control. Turning things on, speeding things up are important, but also important is the ability to slow things down and stop them altogether. Your gas pedal isn't any more important than your brake in your car. So you have to be able to use both if you want to operate a car safely. And that's true enzymatically as much as it is in driving or doing other things in our lives. And so here's just an example of this. And again, I want you to understand that this is feedback mechanism. Citrate, if we're building it up, inhibits glycolysis. So we see a red line here, we see a negative sign. That means that citrate, citric acid, will come up and serve as an inhibitor of glucose. ATP will also inhibit the enzyme. That's the one in purple here. That's a mouthful, phosphofructokinase. Okay, it's a kinase that phosphorylates fructose. Okay, but ATP will inhibit that enzyme. On the other hand, if we have metabolites of ATP, adenosine monophosphate as opposed to adenosine triphosphate, adenosine monophosphate will actually stimulate this. So maybe we have a little ATP that's inhibiting it, but if we have lots of this, the ratio of those two is skewed to this side, the stimulation will cause this to speed up and thus make more ATP. So feedback inhibition, negative feedback pathways, where products can inhibit earlier metabolic steps. These last few slides are merely just review for you. What goes into glycolysis? Glucose. What comes out of it? A little pyruvate, a little ATP, a little NADH. Again, we're not making the NADH, we are reducing the NADH with electrons. What about the inputs of citric acid? We've got some pyruvate that's going in. We're making it into acetyl-CoA, and we're going to make some CO2 right there, remember? A little NADH as well. We're going to do the citric acid cycle, starting with oxaloacetate to make citric acid. We're going to go through those steps. In the process, we'll make a little ATP. We'll make a little bit of CO2, but we'll make a boatload of NADH. We'll reduce a boatload of NAD and a little bit of FADH to have electrons for electron transport. Electrons enter electron transport, and we pump those protons. Okay? Pump those protons, and we make water. So the products of electron transport are oxidized NAD and FAD, reduced oxygen, which is now water, and a movement of protons against its concentration gradient. ATP synthase, we're using that chemiosmosis, that proton motive force. As these things go down, it turns a rotor that literally forces inorganic phosphate onto ADP to make ATP. Again, I think that's super cool. And here's the picture from your book, putting it all together. And then we add on to that negative feedback, like the citric acid up here, ATP feeding back and so forth. Lots of possibilities here. It's all connected. We're trying to keep it as simple and straightforward as possible, but I know we're probably taking you out of your comfort zone a little bit. The key is to work each thing individually, get it down, see how it's connected, what are we making, get it down, work stepwise. Trying to eat the whole pizza usually gives you indigestion, if you know what I mean. More wrap-up slide just showing you the difference between lactic acid fermentation, which is found in mammalian cells, and oxidative phosphorylation. You can see there's a big jump in energy production. Even that said, this is still only about 37 or 38% efficient out of a total of 100, but much more efficient. But on the other hand, glycolysis gives us immediate energy. We can ramp up ATP from glycolysis orders of magnitude faster than we can do that. An order of magnitude is usually a 10-fold jump, and it's at least 20-fold faster. So we can, if we need ATP in a pinch, we can undergo glycolysis. If we're not exercising and we're at rest, which is usually most of the time, surprisingly, we can generate most of that stuff aerobically. And that wraps up chapter 9. It's a busy chapter. The key is go slow, do the steps and do them together. This is the shortcut in many ways. If you shortcut the shortcut, if you don't do the work, it will reflect in lower scores and poorer performance, not only in this class, but downstream. So take your time with it. Let me know if there's anything I can do to assist you. And thank you for your time, and I'll talk to you soon.